Hello, everyone. I want to thank you all for being here. We're excited to have you join us today from both across Australia and across the world. I'm told there are 18 countries represented amongst our audience today, which is wonderful. Um, I want to begin, say by with an acknowledgement of country. So we want to welcome you all today, and we want to begin by paying our respects to the traditional custodians throughout Australia. We pay our respects to their elders, past, present, and emerging, and acknowledge the important role that Aboriginal and Torres Strait Island people continue to play in our community. I am joining you today from Yugara and Turrbal country, which is located in Brisbane, Queensland. For those who haven't met me, my name is Fionn McMillan Webster, and I am the science writer and content strategist at TERN, Australia's Ecosystem Observatory. So, just a little bit about TERN for those who aren't familiar. Um, Before I get to that, I just want to let you know about we've got a little bit of housekeeping. So, please feel free to ask questions throughout the webcast using the Q&A function at the bottom of your screen, and we will answer them towards the end. If you have any technical issues, comment in the chat function, and we'll try to resolve them as soon as we can. And you can also use the chat for contact with other attendees. And we are recording the webinar today so that we can share it with those who couldn't attend at this particular time. So I want to, again, talk to you about TERN. We are a national research infrastructure project funded by the Australian government. We make all the observations and data collections that tell us about what's changing in the environment. We also collect physical samples and develop a range of tools and apps, and our goal is to help researchers study both land and coastal ecosystems to gain a deeper understanding of what is happening, including both the magnitude and the direction of change. When we say infrastructure, we mean our people, our instruments, and the data. They're all covered under the definition of infrastructure. So I would like to now introduce our topic and our speakers today. Across Australia's vast drylands and those of other continents, springs and oases emerge as remarkable lifelines with critical ecological and cultural significance. They serve as refuges for unique flora and fauna found nowhere else on Earth, and they've been essential to human survival, health, and cultural heritage for millennia. Yet these precious ecosystems now rank amongst the most threatened on the planet. Today, we bring together leading experts to explore the multifaceted value of Australia's desert springs and oases, examining both the challenges they face and innovative approaches to their conservation. It's my great pleasure to introduce our speakers. Our first speaker today will be Dr. Renee Rossini, who's an ecologist at the University of Queensland School for the Environment. She's an expert on macroinvertebrates of the critically endangered springs of Australia's Great Artesian Basin. She's going to talk to us today about her research in this space. Our second speaker will be Professor Rod Fansham, who is also from the University of Queensland. He's an internationally recognized expert on spring ecosystems, particularly in arid landscapes. He's also the founder of the Fellowship of the Spring, a global initiative to study and protect the world's oases, which he's going to tell us about. And our third speaker today is Dr. Michelle Guzik, who is an environmental scientist at the University of Adelaide in South Australia, where she leads multidisciplinary collaborative research, which includes the design of environmental DNA, or eDNA, meta-barcoding tools to characterize biodiversity in groundwater-dependent ecosystems. So I'm going to go ahead and hand over to Renee and ask her to turn her camera on so she can begin. And I will catch up with all of you after the talks are finished. Over to you, Renee.
Thanks, Fiona.
I will turn off my camera.
I just need to share my screen. All right, it's a real privilege to be here, and I'm very excited to hear there are lots of people from all over the place, including the broader Fellowship of the Spring that are here. My name's Renee. I'm speaking to everyone today from Larrakia country, near Darwin in northern Australia, but my usual place that holds me and that I live is on the southeast Queensland kind of coastal mountains of Unibarra country. But I also like to pay My deep and humble respects to the Bidjara um, and Ingai peoples who most of the work I'm presenting on today, whose country it was collected on, and who were the custodians of an amazing set of springs that I like to think of as part of my body. They were looking after it long before me. So what I've tried to do is just take a really high view here, and I felt like because I was first, there's a bit of pressure there, but it took knowing that I had Rod and Michelle here, it did mean I could stay away from some areas. But I do feel like there's a little bit of a burden of responsibility to explain the system we're talking about. So within Australia, the Great Artesian Basin covers most of the eastern third of the whole continent, and it's on record as being one of the last large aquifers in the world that it still has a healthy kind of balance of recharge to discharge. Naturally, the Great Artesian Basin, before any kind of colonial interventions were put in place, discharged water out across the semi-arid and arid zone into places that are springs. And this is a drone image of a spring from Edgbaston. So lots of them are actually quite small if you work on springs from other places. At AGR, springs are little tiny fish ponds. But Australia does have representatives of really large springs, like at Irrawandy Dalhousie. The way I wanted to frame springs through all of this is that because they live in these kind of semi-arid and arid contexts, but I find it easy to think of them as inverse islands. So they're islands of wetland in a landscape that's generally very arid. And so most of the things that live there can live in the spring but can't live in the surrounding arid landscape. The story of the springs in Australia is in some ways, in the long term, relatively happy. Aboriginal people's relationship with groundwater was huge and healthy, but colonialism did bring some big problems that have really had intense impacts in the first 200 years. I mostly work at a beautiful spring complex called Biari or Edgbaston, and it's a bit of a jewel in the crown of global wetland biodiversity, which I think Rod will probably talk more about. So what I wanted to focus my talk around was kind of 3 things that I think we can all learn about springs and groundwater systems from Biari, but really focusing on what I've learned over the decade of having the privilege of working there. So the first thing I thought I'd introduce everyone to is that this idea of wetlands as islands in a arid desert kind of multiple scales. So that's a picture of the Great Artesian Basin with all of the spring kind of complexes, what we call them, that emerge from the basin across its whole extent. So they kind of exist as these archipelagos across the whole basin. Within each archipelago, there's then multiple islands, multiple springs, and some have many springs, thousands. Some might only have 1 or 2. We also know, because of some of the more recent work that's been happening, since about the kind of 60s, 70s, 80s that I jumped on the wagon of, that some of these archipelagos in general have more species that live there, and particularly more species that are endemic to either just this particular system, so groundwater fed springs, or just to that particular location. Most of the time, within one of these places, they have their local set of things that occur there. You also see a pattern that individual islands within the archipelago also might have more species than others. So not every spring has the same set of stuff, and not all springs across the entire basin have the same things present. And I think some of the stuff that I feel like I've lent into is that this comes down to a couple of different processes acting at different scales, and those scales dictate the causes of what's happening. Hey, Rod, there you go. um, this is an image of a map of Edgbaston, Biari. You can see each of the little circles is a spring, and the color of the circle is how many of the types of endemic snail that just live on that property and in those springs live in each place. So you can see it's an archipelago. There's many springs in that particular location. That's a single property. And then within those, some have up to 7 species or more, and some springs have absolutely none. So part of the work I do and have done is about trying to figure out why. Why does that happen? Why are there some places more stuff than others? Um, and why is there more stuff in springs? So there's some work that Rod and a bunch of our other colleagues have done in the earlier days showing that springs are really unique in Australian aquatic ecology because they're so permanent. This is water that's been flowing out of the ground, since the inland of Australia was a rainforest. Like, this is an ecosystem that's very unheard of in Australia where most water is boom and bust. But something I guess I've liked to try and challenge is that just because the water's permanent, it doesn't mean that it's homogeneous or stable. So here are some photos and a very complex figure that is put here to drive home that this is the same spring in 2 different seasons. So it expands to kind of double or even quadruple its area, in the cooler months, and then sucks back when it's really, really hot, especially if we don't have any rain. And what that creates is these quite physiological extremes in some parts of the spring. So if you're in the kind of light gray areas on the edge of the spring and you're an animal that lives in the water, we've got measurements that would say that you get exposed to things like pH of a 10 in water that's saltier than the sea and can get as hot as 50 degrees. So it's not necessarily a very stable place to live. And what that means is that those physical conditions create different microhabitats across the spring. These are 2 pictures for an example of 2 extremes. You've got these nice, deep, permanent places that stay pretty much stable all the time, and these shallow, impermanent places on the edges. And what we've found happens is that I'm a snail person, I mostly work on the snail assemblage, is that those gradients suit different species more than others. So Glyptophysa, in this little map here, you can see it's only ever found right in the core of the spring where the conditions are very stable, compared to another species from a different family, Gabia fontana, which is far more tolerant and spends a lot of its time actually preferably out on the edges. For most of these species, they're stuck in those particular places, because what we've found is that it's actually lethal for them to live outside of it. These are species who taxonomically come from groups that are generally rainforest species. So there's not been a point in their life where they ever adapted to these kinds of conditions. So what happens if the chemical conditions change, either at a microhabitat or at a whole spring, is that those species can actually go locally extinct or extirpate. So this is some results from some experimental work we did, that we found that if you expose Glyptophysa to the conditions that are common in the tail of a spring, most of them die within about a month. So if a spring changes, for them, they're not going to last much longer. And I guess at a system level scale, what that means is that not all species occupy every spring. This table is showing you the occupied percentage at Biari where these species live. But frequently, this is across 5-year timescales, we're experiencing extirpations and colonizations pretty constantly. So this is a really changeable system. But what is keeping it healthy is the fact that these animals can move around. The last part, and that's what I thought I'd finish off with, is that, obviously with something that's so changeable and animals that are quite sensitive and plants, we run the risk of extinction. The Greater Teaching Basin is at a national level listed as a threatened ecological community, and that's across the whole basin. We've lost enough across the whole basin that we're really worried about it as a whole. So some of my work, and particularly, the longer I work, I get really more obsessed with the conservation outcomes that we need to be fostering for springs. Something about Australian springs, from the kind of white science perspective is, really thorough kind of scientific discovery in the system only really started in earnest in the 70s and 80s. So we've got a big gap to fill. And what that has created is a big data deficiency across a very diverse system. So these red and green and yellow plots that are there to try and summarize, or they come from papers where we summarize some of the conservation conundrums for the system. And one is that for most of the basic information we need for these taxa, we don't have any published literature and there's no one working on them. But if you use the information we have and you assess them against the broader criteria, most species from the GAB system, that figure is just for the gastropods, should be listed at some form of threatened, most of them critically endangered if we use our national legislation's criteria. The picture on the bottom too is that they are endangered for legitimate reasons. So that's a line for every single springs complex. And if you look at where the threats that we know cause declines in these taxa are occurring, some of the most diverse places are being exposed to all of the threats, and nearly all of the taxa there are highly susceptible. But I didn't want to end on a bad note. So I think another beautiful thing about working at Biari is that we work with the red-finned blue eye. It's the most famous of the GAB springs assemblage. It gets the most media attention. And that's because it's a real Aussie battler kind of story. When Bush Heritage, the conservation organization that managed the property, now acquired it, and before, when people like Rod and Winston Ponder were working on these systems, the red-finned blue eye was on a very strong trajectory towards extinction. But what this graph is showing is there's a lot of data that goes behind that, but from 2021 especially, with some really huge investment and some really strong conservation action, including captive breeding and really strategic captive breeding to rebuild genetic diversity. We've brought this species back onto a really positive trajectory, and it's at the point where we could almost take it down a listing from critically endangered to endangered. I just put a lot of little video in here of the red for everyone if it works. So they're really a great case study of this system is incredibly diverse and unique. It's facing a lot of problems, but there's perfect examples of where when people kind of knuckled in and worked together, we've really turned around the trajectory for some of the really threatened species. If you want to know more, there's lots of things you can do. This is a real speed view of Edge Bass, and there's a lot about the springs you can learn. So feel free to reach out to me at my Bush Heritage email. If you just Google the red-finned blue eye, there's a lot of really great media, including really good video and photo content. And if you want to read more about the reserve as a whole, head on over to the Bush Heritage website. That's it for me. And now I'm very lucky to pass over to Rod.
Brilliant, Renee. Thank you. And I need to share the screen. That one. Well, thanks for having me here today. And I'm following on from Renee. We had a little chat about the vision of what we wanted to talk about. So there's not a great deal of overlap, but we start by revisiting the Great Artesian Basin. Here it is again. The water enters the basin from the elevated areas on the eastern side, which is depicted here in dark yellow, and travels on its merry way underground in a confined system and discharges in those red envelopes, which are where these spring supergroups occur that Renee talked about. Now, a confined aquifer system, you can think of that like a pressurized balloon full of water squashed by a blanket. So the pressure comes from the blanket, and also the pressure comes from the elevated areas on the edge that provide the hydraulic gradient. And the springs will pop out where they can in this pressurized system. The green's the blanket. The blue here is the pressurized balloon. And the important thing for us and for these islands in the desert is that the water can travel an awful long way from where it goes in to where it comes out, and it can migrate across climatic gradients. So it can go in high rainfall areas and come out in a desert. And it can come out in a range of situations, but this is around the base where the basement is elevated, creating a sort of island in the middle of the aquifer, and it'll leak out around the edge. These are granite basements down in southern Queensland near Ullo. And it can also come out through faults. And here's the geological fault in the bottom right, the Burke River fault, and the water's coming out feeding a well-known spring near Bulliott called Elizabeth Springs. So this pressure, the springs they're advertising their own demise, really. There was obviously water down there, and it wasn't long before Europeans got inquisitive about the nature of it and started drilling holes. And they penetrated the blanket and got through to the balloon. And to their great jubilation, the water came out under massive amounts of pressure because of the elevated intake beds. You could have, up to 1,000 meters of pressure, which is an awful lot of pressure. And the pressure was so great that the discharge volumes were enormous, and they directed that water down artificial drains. And before long, there was thousands of these pinpricks in the balloon, and thousands of kilometres of drains weaving their way around the landscape, effectively watering the drier parts of Queensland. And it's a primary reason why Queensland's such a rich pastoral economy, because we've got this unbelievable water resource under the surface. All you've got to do is drill a hole out. You don't have to pump it out. It comes under pressure. And my travels around the world made me realise just how unique this system is. So with the loss of pressure, with digging all those holes, the springs, many of the springs disappeared. So this is the town of Yantaboola, which is a ghost town. We get, this map shows, great aspirations that were never realised because the springs went extinct that were going to provide the water supply for the town, and the last resident left in 1980. That's the little town of Yantaboola in northern New South Wales. You'll still find it on maps. So the signature of the extinct springs, the inactive springs, are still there because they leave these sort of sulfate residues behind. So they're relatively easy to map, but they're also well recorded on historical sources because they are so important for the early years of pastoral development. And we've put a lot of work into that and quantified not only the active springs, but the inactive ones. And it turns out about 50% of the springs are either totally extinct or partially inactive as a result of loss of aquifer pressure. So this exploitation of the aquifer through bores is undoubtedly one of the major threats for this, or the major threat for the springs. But there are other nasty things we do to them as well. As a result of the desire, the requirement to use some of that vast underground water resource in the Australian mining industry, there was programs to start to use the water more efficiently, basically putting those open pressurized bores into big taps and then directing the water through polythene pipe rather than open drains where most of the water evaporates. And this commenced with a sort of mining initiative to recover water for the Olympic Dam mine in South Australia. And then it progressed into a sort of public funded thing that had various iterations. It's still going, actually. And it's been reasonably successful at restoring water, a big public initiative where matching funds between the private and the public sector cap these bores, do this big engineering job. So we've been interested in what the consequences of that, not only the loss of pressure, but also the gain of pressure have been. And on this map are a bunch of bores with different colours. You don't have to worry about the colours too much, but they're the bores around Pelican Creek, which is another name for the Biari Edgbaston Spring Complex that Renee was talking about. Really, really significant high value place in terms of biological values of springs. So this is all the bores within 50 k's of that site. And this bottom graph here is what's happened to them. So they're progressively getting put in, peaking in about 1950 in number. And they're just open. The blue is open flowing bores, discharging huge volumes of water through each one of those holes. And this is GABSI in action, gradually putting them into pipe And you see in this region, we've done a pretty good job. And there are pressure measurements on the bores. And you can think about a pressure measurement as the height with which the water would extend in a vertical pipe without friction. So out in this part of the world, they had about 20 meters of pressure, awful lot of pressure. And that pressure was lost by about 1960. And then with GAPSI, we've regained about 6 meters of pressure. And that's going to be an ongoing process as the lag time between the pressure increase and the bore capping is made up. Consequences for the springs. This is again the Biari complex, the Pelican Creek, that Renee flashed up. These are the springs, and this is the total wetland area, which is a surrogate for the discharge. So the more water coming out of the vent of the spring, the bigger the wetlands. So we can relate the size of the wetlands to the amount of water discharging. And this is 12 years of monitoring the wetlands from 2008 to 2020, and you can see they've recovered. We were pretty convinced that was because of pressure recovery, but it might have been rainfall. This is the rainfall, it's a wet period, but the recoveries continued through a dry period as well. So it's undoubtedly a result of restoring aquifer in that region. Okay, so Renee's touched on our journey with biological discovery, which has been an absolute thrill as a biologist. It's also been thrilling to have a bit of an impact on public policy and recovery of springs. And so with that in mind, I couldn't help thinking a few years ago when I had time to think about, reflect on my career and what I wanted to do. I couldn't help wondering whether there might be people in the rest of the world who I could coordinate and see if they have the same passion that we have for springs and the same excitement about discovery and conservation. And if you think about springs in a desert as geographic sort of entities, they're really powerful entities. You know, they have a sort of corresponding mythology through across different cultures. The endemic values that we know of for the Great Artesian Basin are also repeated in different places, and the story of their demise and hopefully potential recovery could also be repeating around the world. So I set about pulling together everything we know about springs in the drylands of the world, which is the part with less than 500 millimeters rainfall, depicted in pale yellow on this map. And I just trawled the literature over many, many, many lonely nights and organised the data to make a map of springs across these areas and coordinate the published literature in relation to keywords and topics. So we now have a reasonable understanding of what we know about springs around the world. But perhaps more importantly, I entered into a sort of form of academic internet dating by corresponding with people that I came across who'd written these articles. And very often they ignored me, but sometimes they responded. And they were indeed absolutely enthusiastic and with this common understanding of the importance of springs. It was like meeting long lost relatives when I finally got to talk to them. And the group has grown from small beginnings to 48 people from 23 countries. And I've had the good fortune to visit many of these people and have the ambition to extend the networks, not just sort of from me, but also to extend the networks between all the people in the Fellowship of the Spring, and indeed to expand the Fellowship of the Spring. Our ambition is to just basically help each other, and particularly help people in disadvantaged and less privileged countries in Australia to get work done and to go on their own journeys in their own way that might parallel the sort of exciting journeys that Renee and I have had here in Australia. And indeed, it's working. We now have these networks happening, but indicated by all these arrows around the world, of people who are working together on different projects across the continents and across the countries. And I'm just going to talk about two of those wonderful people. First one's Dr. Ravi Asaman, who's in Tunisia. I have a good fortune to travel with her last year. And she is totally passionate about springs, extremely hardworking, diligent, smart. And she took me around and we saw many springs in southern Tunisia and she, with just a few scraps of money that I've been able to give Ralia, is going to continue that work and document the story for the springs in her part of the world, which unfortunately is a pretty sad story. Nearly all the springs have gone extinct in the last 30 years within the living memory of the people we spoke to as we pulled up on the side of these dusty little oasis towns. The year before, I went to Ethiopia and teamed up with Ali Ahmed Bila, and Ali's a young anthropologist in Samarra, which is the capital city of the Afar province of Ethiopia, which is one of the poorest, driest, hottest places on the planet. The Afar are nomadic pastoralists, and we documented what it means to have a permanent, perennial, productive resource, aka a spring, on the estate of a nomadic pastoralist. And the story we told was of the absolute hospitality of people in the Afar for their own people. When they run out of resources on their country, they're always welcome on your country, but also the important resources around a perennial spring mean that unwanted outsiders, mostly Somali people, will invade and actually kill people. And this has happened in recent times. So they're actually the site of pretty intense conflict, as well as sort of the oasis paradise that we have in our mind's eye. So I'm really hopeful that we can extend this network, build on our work, and get really good things happening around the world with the cross-fertilization of all our skills. And at the moment, we don't have any funding. So if there's anyone out there who wants to help us, feel free to contact me. Thank you.
So, hi, I'm Michelle Guzik. I'm based at the University of Adelaide. And I'd just like to acknowledge that I'm presenting from Kaurna country of the Adelaide Plains. But I'd also like to acknowledge the Arabana people, who are the traditional custodians of the region of springs, that a lot of this research was undertaken. It's been my privilege to make an attempt to improve our knowledge of springs and the animals that live within the springs, and also to gain some understanding of the fundamental connection that Indigenous people have to water and specifically springs. So yes. So on to my research that I'm going to present today, which I've entitled Using Environmental DNA in Biodiversity Assessment and Management of Great Artesian Basin Springs. And this is a collaborative research program undertaken by myself, but also a really high-level, critical collaborative team, including Nicole White from Curtin Uni, Perry Beasley Hall from Adelaide Uni, Mika van der Heide from Curtin Uni, Nick Murphy from La Trobe, Rachel King from South Australian Museum, and Danielle Stringer from Adelaide Uni. These people are experts in their fields of eDNA, taxonomy, systematics, phylogenetics, and without them, we couldn't have progressed as much as we have. So just wanted to acknowledge those people. And so over our collaborative research has sort of happened over the last 20 plus years. And in our research, we've established exceptional levels of endemism in Great Artesian Basin springs in South Australia specifically. So Rod and Renee were talking about Queensland springs, and we've focused on the South Australian springs. And what we've discovered in our phylogeographic and phylogenetic research is ancient connections of invertebrates across arid Australia, but also within springs, the endemicity and the genetic differentiation between springs is really high. So historically, it was assumed that the invertebrate endemics were a single species that was spread throughout all springs, including the Queensland and New South Wales springs and the South Australian springs. However, our research has shown that there are many more species and that they are endemic to particular short ranges, short geographic ranges throughout the springs. So they're quite unique. And we did an inventory of South Australian springs and put together all of the animals and plants living in and around springs. Recently, we published it in a paper by Perry Beasley Hall, and we also published online the full list of species that are in and around springs. So our work has highlighted the uniqueness of the springs and the short range endemics that live there. With that comes new species, and we've been fortunate enough to have expertise with Rachel King and Danielle Stringer describing new crustacean species, because we recognise that if animals from springs are to be recognised under legislation and policy, that they really need to be described formally. And so because taxonomy is quite slow, we've progressively described species over the last 20 years, and we've been quite fortunate in being able to do that. And so a fairly recent publication by Danielle Stringer described new isopod species that live on the margins of springs in South Australia. But we also recognise that there are fundamental cultural and environmental concerns for spring survival. And so Rod and Renee have identified that, and in collaboration with Arabana Aboriginal Corporation, who are the traditional owners of the region specifically that we've worked in, we understand their concerns with extinction of some springs in that region. And we expanded on that a little bit in a recent paper in Frontiers in Environmental Science. So these concerns are real, and we know that we need the best available information to be able to try and monitor and understand fluctuations in endemic populations. And so basically, the basis of all of our research is that knowledge, evidence, and uncertainty determine our confidence in regulatory decision making, and any improvements in evidence and confidence in survey data will improve environmental conservation outcomes. And so we've worked towards that with our by using new technologies. And so our research program is developing cost-effective methods that will detect groundwater-dependent fauna without physically catching them. I'm just realising that I might have that thing there. Sorry. That detect groundwater-dependent fauna without physically catching them, that we aim to develop methods that are quick, effective, and repeatable for surveys and to improve decision-making capability. And so we're employing new DNA sequencing technology that allows us to do that in a way that we've not really been able to do before. So we're using next generation DNA sequencing to sequence and characterize environmental DNA. So that we're all on the same page about what environmental DNA is, because it's the kind of basis of the rest of the talk, I'm just going to do a quick definition so that we're all on the same page. So environmental DNA, or eDNA, is DNA that's shed from all organisms into their environment. It comes from the organisms from hair, scales, skin, waste products, reproductive cells, and it's free DNA in the environment. And on the principle of this, if we're able to DNA sequence the eDNA, then we're in principle able to identify the animals living in an environment without physically catching them if we take a sample of that environment. So an environmental sample can be a water sample or a soil sample or an air sample. And in principle, if there is eDNA in that sample, when we DNA sequence, we're actually DNA sequencing the animals living in that environment at the time of sampling. And so we armed with this kind of knowledge that's basically on the basis of research that's been done in marine and surface freshwater environments, we're aiming to apply eDNA sequencing into groundwater. But as we all recognise, groundwater has quite unique properties compared to surface and freshwater environments. And also, the organisms that live in groundwater are quite unique with the short-range endemics, very ancient evolutionary timescales, and not very well known or characterised at all. Most of the fauna are unknown to science. And so when we consider that we're trying to DNA sequence very tiny fragments of DNA that are continuously breaking down in the environment, there's basically, molecules of DNA, and they're breaking down all of the time. Our methods are essentially developing, we're basically looking for a needle in a haystack. We're trying to develop molecular magnets that will tease out fragments of DNA that we can then sequence out of an environmental sample that's got lots and lots of different things in it. So that's essentially what we're aiming to do with this kind of technology. So overall, our aim is to improve biomonitoring of endemic fauna from groundwater-dependent ecosystems using eDNA technologies. And we have developed, because we're research scientists, we develop, we go where the money is, essentially. We obtain research funding that will allow us to undertake the fundamental science that we can then apply to hopefully any groundwater environment. And so we've been fortunate enough to have quite a large 4-year grant to do the proof of concept research for groundwater dependent ecosystems in the Pilbara. And so I'm going to talk a little bit about that, but we've also then, our aim is then to apply those methods to other groundwater dependent ecosystems like the springs. And we have a consortium of researchers that we're working with at the moment under the Greg project. So I've just got the URL on the little laptop there, where we're conducting a global eDNA study of groundwater dependent ecosystems. So that's something that we're hoping to expand in the future. And so I'm just going to now talk about the groundwater research that we undertook in the Pilbara as the proof of concept, and then talk about the Great Artesian Basin Springs and what we've done there. So our initial proof of concept was that we developed the molecular magnets to detect a single species that we know exists in subterranean groundwater aquifers in the Pilbara, the blind cave eel. It was known from only a couple of locations in the Pilbara prior to our research. And once we developed our assay or molecular magnet, we were able to take groundwater samples, and we were able to apply the method of analysis, and we extended the range of the species by about 3 kilometres. But since then, our method's being applied in surveys across the Pilbara, and the range of that species has been expanded quite substantially, essentially just taking groundwater samples rather than physically sampling the animal. So that's quite unique and was really important for us in terms of demonstrating that we can do the work. Apologies. And then we've also undertaken community-wide detection of organisms living in groundwater-dependent ecosystems. So these are subterranean species. And in this graph, what you can see is that we used published eDNA molecular magnets for detecting similar types of communities in marine and freshwater environments, and we applied them to groundwater. And we were able to show that we could detect quite a number of the animals that we know live in groundwater at those particular locations. But we also showed that compared to physically catching animals, sometimes we were not able to detect using eDNA alone. So as a proof of concept, this was really important, and this approach was applied in surveys is now being applied across surveys in the Pilbara. But there is still a lot of work to be done to get these molecular magnets to where we want them to be consistent and repeatable in detecting all of the animals that we're interested in from groundwater. And then, in addition to the methods for analysing the eDNA from groundwater, we're also developing databases of DNA sequences as references. What we know is that when we DNA sequence a groundwater sample, we're basically sequencing a DNA soup. There's DNA from every organism that lives in that environment. And so every sequence is anonymous. The reality is that we need to query the anonymous DNA sequences that we gain from the sequencing to a reference library of known sequences that are paired with a known species. But of course, as we all know, for groundwater-dependent ecosystems, most species are unknown to science and not described. But we still think we still need to develop these local databases to be able to get a handle on what is in the eDNA sample. And so that's something that we've been working towards, and we've undertaken this for the Pilbara, but we also aim to also do this for the Great Artesian Basin Springs. And we've recently published the Pilbara Barcode Reference Library as well. And so then our data from springs, so we undertook a smallish pilot study where we sampled groundwater from bores, Great Artesian Basin bores, and also Great Artesian Basin springs, and using well-known molecular magnets, we were able to demonstrate that when we compare springs with bores, for metazoa, the diversity of the metazoan diversity of bores is quite low compared to that of springs. So we only have a couple of taxa that we're detecting in bores, but we are detecting quite a large number of different invertebrates from the springs, including snails, crustaceans, and insects, which is what we expect. I'm not going to go into the details of distributions and what we've detected where. This is unpublished data, but it's giving us an indication that the approach works at springs and that there is scope for development, which is really important. So future research on springs will include development of taxon-specific assays, so molecular magnets that will allow us to detect particular species, because under state and Commonwealth legislation, especially Commonwealth legislation, certain species are of interest over others, partly because they are listed under the EPBC Act. So that includes snails, but but we also want to understand community-wide differences between springs using eDNA, and so that will be another aspect. And we'll also be we would also need to curate a local DNA reference library, so a spring-wide DNA reference library that will allow us to query our eDNA sequence data against to identify what's living in the springs. And so I just would like to say a thank you for funding for the spring work specifically, from BHP for the eDNA for Global Environmental Studies, or EDGES program at Curtin University. Also contract research project that was funded by BHT at Adelaide Uni. And also our collaborators, Arabana Aboriginal Corporation and Jake Thornhill for filtering water samples, and Andy Austin for earlier research on Great Artesian Basin springs. And that is the end.
Thank you so much, Michelle, and thank you to Rod and Renee as well. That was all really interesting, and I know we've had a couple questions come through, so I will just share my screen again. And sorry, there we go. So just had a question come through, and I'll just bring that up. In the meantime, I'm finding that one. A question from Renee.
It's one too, Fiona, that I answered, but I make sure you can see it. It's about Perny boar.
Pardon?
There's a question about Perny boar as well that I saw it puts it in answered just because I.
Okay, wonderful. Thank you. Well, actually, a question for you is, do we see invasive species spread within and between the archipelagos? And if so, how? Or do they get kind of limited by the variety of ecosystems and habitats that are there?
It's a good one. Rod can probably talk to plants. I can talk to plants too, but one of the, I guess, two of the most problematic invasives that we deal with at apiary, Edgbaston, especially when it comes to redfins, are the invasive mosquito fish. And most of the time that gets into the springs from other arid zone waterways. So any springs system that connects at some point during flooding into an already invaded waterway is at risk. But luckily, so far, they haven't actually got into many springs. Piari is one of the few. The other is cane toads. You would think, being an amphibian, that them moving around between springs would actually be quite hard, but they're making their way all the way across to the Kimberley now and impacting the mountain springs there. So fauna-wise, I guess another one that's really problematic, not aquatic, but is pigs and anything that causes hooved animals that cause disruption to wetlands. Again, they're not hampered by needing islands of wetland in an arid landscape. They're pretty good dispersers. So unfortunately, I find, at least in my thinking, most of the impacts that you expect for an arid wetland are occurring in the springs. Plant-wise, I don't know, Rod, you got any?
I'll just mention the paragraphs at Edgbaston. So, getting rid of invasive plant species is usually a pretty frustrating exercise because they just come back. But in these little archipelago islands in the desert, you got a chance of beating them. And very satisfying few days with a bunch of students pulling out paragraphs from a couple of springs at the area that's never come back. So,
Yeah.
Cool.
And for biodiversity monitoring and receiving environmental impact monitoring, this is a question that's come through. Would you expect eDNA sampling to replace Oz Rivers sampling and methodology, especially in highly variable environments we see in North Australia? I imagine this one must be for Michelle.
Good Michelle question.
Yeah, it's a really good question, and it's something that we are, we're quite focused on. There is still a lot of work to do with eDNA. I'm not sure if it's come through. We've done a lot of work, but there's still a lot of work to be done to fine-tune the methods specifically for groundwater because there are so many variables. And research funding limits how long and how much research you can do. So we definitely have a goal that we would then want to, beyond just getting a snapshot of what's there on the day that you were there, you would also want then repeat sampling over time. And we just don't have a handle on temporal changes with eDNA yet. And even in the marine and freshwater surface environments, that's still, still being worked out. But I would say that eDNA tools currently are complementary, and they should be complementary to any traditional surveying and sampling. And I don't think that would change for some time at this stage. There's just so much that needs to be done.
Can the eDNA suit, and this is sort of a follow-on question by a different audience member, can the eDNA suit be ranked for diversity at least, so that in the absence of identifying individual species, we can still track trends in diversity?
Yeah, and that's an area of research that is much better developed in other ecosystem types, but even the microbial communities and the fungal communities are that kind of work has been done a lot. And I think that in the absence of taxonomy in the future, there will be, we will be using eDNA as an indicator of I don't want to say water quality, but that we will have an idea of diversity and then, potentially health. But you don't want to, I don't want to jump the gun there, right? Like there's still so much that we need to learn, but yes.
And thank you so much for that. And I guess we've just had another question come through, which is, and this could be open to any of you. Can you talk about the reciprocal knowledge sharing with the local custodians?
I feel like all of us have our own story here, right? Maybe Michelle, do you want to keep going because you were last and we'll work backwards?
Yeah, so I think for me, maybe in some contrast to Rod and Renee, I think I come from from a university perspective. I work within the university. I go out and I do field work. I engage at that level. But more recently, I've been engaging or collaborating with traditional owners much more from a less traditional standpoint where, I talk to them about what they need first. And so it's a slow process and it's not on my terms, right? So learning and understanding is not my right. And so I only gain from it what they have to offer. But we've had really positive collaboration, and the kind of the difficulties that we have with industry, academia, and traditional owners, and these springs that are so sensitive environmentally, are things that probably we can all work together. And that's what I hope to do, is to sort of bring the community together. But that just takes time. It's a trust-based relationship. So that's my experience, but it's probably quite different to Rod and Renee.
Okay, John.
For that. And yes, Rod and Renee, if you could weigh on in this. Oh, unfortunately, I only have a little bit of time left, but this is a very important question, so I just want to get your take on it.
Renee, if you want to go.
Oh, I mean, I guess I think what a great thing about Rod's work, would be good for you to touch on, though, is just expanding into Indigenous communities across the world. It's not just Australia. Um, but at least for me, I guess I just felt so lucky every time that I've gone to springs places, like Rod said, and I think like lots of us would say, that they are nodes of connection and concentration, and they are often very special places. So I've had nothing but amazing connections with people who've been willing to share stories. And I think it also, from a biodiversity conservation standpoint, for me, it really can help you change your perception of what value is. There's some really good examples from where, closer down near where Michelle works in South Australia, about how walking two ways and managing springs together doesn't always mean that it's about getting rid of a plant that's considered invasive. You have to walk that path together and talk together about making decisions.
Um, I'll give a plug for TERN here. One of the things we did was, make our database public access, so people will get online and find out about these things. And I've had two really meaningful exchanges with Aboriginal people who found the name of a spring that they remember from stories from a relative. And this is Queensland, where connections to culture are terribly fragmented, but it's also a testament to the power of the icon of the spring. That the stories around the springs are preserved even as fragments, and can be so meaningful for people. And they find this name that they remember a lost auntie talking about, and they've contacted us, and we've been able to take people back onto country to those sites and meet the current landholders. And it's just been fabulous. Yeah.
Thank you all so much for that, and for all of the talks. It's been absolutely fascinating. I just want to let the audience know that we will have more webinars, obviously, in 2026, bi-monthly. The next one will be on the 1st of April, where we look at nature's litter, nature's treasure. We're looking at the decomposition of leaf litter and biomass and its importance in ecosystems and carbon cycles. And look, once again, I just want to really thank the speakers, as well as all the audience members who came to attend today. We had a wonderful turnout. I'm not surprised, because this is a really interesting topic. And I'm looking forward to hearing more feedback and wanting to talk to these speakers. Please follow their work and keep in touch. Thank you so much to everyone.